those chapters we will only tackle for entrances, like things which are asked in entrance. Because if they are eliminated from CBSE board, so you will not be quizzed on those chapters in your school or in the boards, right? But still there are certain concepts that can be asked in the NEET or the entrances that you want to give. So that will be like, I will decide what portions have been asked in entrances before, and we'll do that. Okay, Fatima?
Okay, sir.
Yeah, okay. And would you like to suggest something otherwise? So this is what
No, sir, I was
planned.
asking because I was planning on giving the NEET, that's why.
Yes,
So
right.
I wanted to know.
Yes, of course. So even for NEET, I've told you that they say that mostly NEET, 98% it comes from NCERT. It does not go beyond NCERT, but still there are some concepts which are not properly explained in NCERT, but they ask questions on that basis. So that I anyways, from my side, we have to do that, right? Because one component of these classes is to also prepare you for entrances, not just for NCERT or board exams, correct? So we'll do the components which are important for boards, which are important for NEET and entrances, okay?
Yes, sir.
Yeah, okay. So if no one has any doubt or question to begin with, then I would like if someone can give me a quick summary. Anyone who would like to volunteer for a quick summary? No one would like to volunteer? I mean, by now I think everyone should be fighting to summarize. So I think you all should need to get out this inhibition. Anyways, if no one will volunteer, then I will have to pick someone until someone agrees, because you also have the right to say no. So that's always there. So let me start with Hanifa. Hanifa, are you there? Would you like to summarize today the last class from here?
Yes,
Okay. Yes, that's great. Oh wow, thank you so much, Hanifa. Let's go. Whenever you are ready.
So in the last class, we had studied about the genetic material, which is the DNA, and the plasmid, which is a circular DNA. It is a self-generated, venomously replicating DNA. And then we talked about gene cloning, and we talked about restriction enzymes, which restriction enzymes are enzymes which are used to cut open a DNA.
But why, just one question, but why all of a sudden we became interested in studying DNA, genetic and plasmid? We started focusing on plasmid being circular. Why?
It's genetic material.
Yeah, but there is a genetic material. Plasmid is not the genetic material. It is just an extra chromosomal circular DNA. Why are we interested in plasmid? So if I'm asking questions, it's not for only the person to whom I'm asking question. Anyone who would like to answer can just unmute and answer. Remember, keep it as a discussion forum, okay? Instead of me taking your viva or something like that. Why we are interested in plasmid? Anyone? We are interested in plasmid because it has a lot of scope to be used as a vector DNA. It's small, it replicates on its own, and it's a very good thing. We can just create a nick in it, put our DNA of interest, and we can send it inside a bacteria, because anyways, it is well accepted by the bacteria and it replicates inside a bacteria. So that's why we are interested in plasmids. And I told you here that the help of, okay, let Hanifa tell this. Yes, Hanifa, continue.
When we learn about restriction enzymes, restriction enzymes are enzymes that use to cut open a DNA.
Mhm.
Then we talk about the origin of replication, which in chromosomes it's a specific area where the replication starts. It represents a sequence.
Yes.
And we talk about why DNA is circular because there is no free and where an exponent can act.
Sorry, I could not hear that part. Can you be a little loud, Hanifa? Yes. Can you say what you just said? I missed it actually. Yeah.
The DNA is circular because there is no free end where exonucleus can act. That's why it's circular.
No, plasmid is in general circular. It's not circular for a reason. It is a circular DNA, and that is where we utilize that property of that DNA. It's like why is chlorophyll green? It is green, that's why everything else happens. So plasmid DNA is circular, and genetic DNA might not be circular in many organisms where it's in the form of a chromosome. Okay, so it's not circular because so that it cannot get cleaved. Because it is circular, it cannot easily get degraded. It's stable. Okay.
Yes, sir. And then we talked about tools for recombinant DNA technology, and there are two types of restriction enzymes: exonuclease and endonuclease. Exonuclease digests DNA from the end, and endonuclease can identify a sequence and cut it there.
Yes, correct.
And there are two types of restriction endonuclease. One is E. coli and Hind.
They're not two types of, they are just two examples I have taken from. I told you that we have been able, like scientists have been able to find more than 900 plus restriction endonucleases from around 250 bacterial strains. So these are two examples that I gave to explain how these endonucleases are named. Right, so what eco R1, these two are in your textbook as well, so that's why I chose these two. There are many others, you know, there are many other kind of endonucleases like BAM-H1, SAL1, PVU1, PST1, PST2, and many CLA1, which is called. But how are all of them named was the point, which I was trying to explain, taking these two as an example. Okay, don't think that there are two types. They are not types. Is it clear, everyone?
Yes.
Yeah. Yes, continue.
They are named on the basis of the source of origin, the strain, the original strain, and the order of isolation.
So the first alphabet is always capital, and that first alphabet with the successive another alphabet or so tells us about the source organism. So eco here is for E. coli. Okay, R is for the strain of the E. coli from which this endonuclease has been isolated, and 1 is the order of isolation. So if it was eco R2, it would have been the second enzyme that is isolated from the same eco R bacteria and its strain. Okay, 1 means it was the first to be isolated. Similarly, you see Hind2. In Hind2, the alphabets HIN tells you about the source, which is Haemophilus influenza. H for Haemophilus, IN for influenza. The word D tells you about the strain, which is the DS strain in this case, and 2, the Roman 2, tells you about the order of isolation. So it is the second endonuclease that has been isolated from the same virus of the same strain. So there must be a Hind1 as well. Correct?
Mhm.
Yeah. Okay. Continue. Then, oh, I think we
And
ended.
then,
Yeah. Tell me.
and then finally, we talk about the sequence which restriction under the stabilizers, which is a palindromic sequence.
Yes, palindromic sequence. What is a palindromic sequence? Did I tell you about this?
It's read the same word either of the side.
Yes, it's read the same when you read it in different directions, like different strands. So, for example, a word I always use, which is Malayalam. So if you read it this way, it reads the same Malayalam. If you read it this way, it's the same Malayalam, right? So the word Malayalam is like a palindrome, correct? So similarly, in this case, if you see, if you read it from 5' to 3', like on this strand, it is GAATTC. If you read it this way, it's again GAATTC. Or if you read this strand this way, it's CTTAAG. If you read this strand this way, CTTAAG, correct? So you understood that why it's a palindromic sequence? Yep. Okay, thank you so much, Nifa. And I will take forward from here. So as we all know now that restriction endonucleases, all restriction endonucleases have specific sites, and these sites can be 4, 6, 8, or whatever base pair long, but they have to be a palindromic sequence, right? They bind to a palindromic sequence, and the reason is that how these restriction enzymes cut DNA is what is of importance. It is very important to know. So they do not cut just a, they do not just make a blunt cutting like anywhere. So they will always cut between the same two adjacent bases. For example, in this, if on this strand they are cutting between A and A of this palindromic sequence, on the other strand also they will cut between A and A only. And the benefit of this is the cutting will be like this. It will not be very smooth. It will be blunt. It will leave some sticky ends. So for example, this TT will TTA and this ATT, they will stay flanking. They will stay open like a sticky end. So if you use the same restriction enzyme to cut your DNA of interest and also to cut the plasmid vector, there will be the same kind of sticky ends present in both. And then it becomes easier to just make them bind together using ligase. Is it clear, everyone? You understood what is a sticky end? Yes, no? No one is speaking. No one is speaking, I think. Okay. I'm not sure if you understood, but since no one is speaking, so I'll move forward. Now, once we have cut the DNA successfully using restriction enzymes, what do we do? What do we want to do next? So let's go to the next point. What do you want to do now? You have cut the DNA. What now? You made many fragments in your DNA. Now the next logical step is to figure out which is your fragment of interest, right? And because you have designed the experiment and you know which restriction endonuclease you have used, so you will also know, because of you will also have the information of the size of your gene, right? The gene fragment. So the next step is separation. Okay, separation and isolation of your DNA of interest. DNA fragments. So the DNA of the fragment of our interest, which we want to use further to clone and to do biotechnological processing, how will we separate it? So we can separate it using a technique known as Agarose gel electrophoresis. Anyone know what is this? In this chapter, so how many of you have this chapter completed in your school already, or is it going on? Donna?
Yes, sir.
Done?
It's done.
It's done. Okay, so I expect some contributions and discussions from your side, because then it has to be the second time you are hearing these words, right? Fatima, Hiba, Rifat and Atif.
It's not started in my school.
Not started yet. Okay, Hiba, Rifat and Atif.
That's going on.
It's going on in Hiba's school. Okay. Others?
That is going on.
It's going on. Okay. And Rifat?
Done.
Done. Okay. So I think everyone, I will not expect from Fatima, but if she contributes, because based on her reading, that will be great. But from others, I at least expect, to answer. So did you read about Agarose gel electrophoresis? Anything? It has 3 terms. Let's go term by term. Oh, I'm so sorry. Okay. It has 3 terms. One is agarose. What is agarose? It's a biological polymer. Okay. And where do we find agarose from? We extract it from a seaweed. Okay. Seaweed is a plant, aquatic one. We get agarose from there. Okay, cool. It's a biological polymer, just like any other biological polymer. But the good thing about agarose is that it can be converted into a gel-like structure. Gel means it can be solidified, right?
Yes, sir.
But not completely. It's semi-solidified. And how it works is, so let's say these are the fibers. These are agarose fibers. I'm just randomly using a concentration and making all of them come together and become a semi-solid gel-like network. Now look at these empty spaces. So let's say this is one concentration. Let's say this is 1x concentration of this polymer. So when you will make them come together and become solidified, there will be spaces left, right? In between. Can you see these spaces?
Yes, sir.
Yeah. So when they solidify, there are spaces left like sponge. Correct? You may not be able to see it with your naked eye because they are very, very small, but I'm just making or trying to highlight some of them so that you can make a quantitative assessment. So look at this size. Now let's say I take too many polymer, like I take a higher concentration. So there are too many polymers and it's very dense when it is becoming a gel. So if there are too many polymers running here and there from everywhere, this is a higher concentration, right? They're all intertwined together. Let's say this is 3 times the concentration. Now look at the pore size. Can you find any difference in the pore size now when the concentration is more?
Smaller.
Yes. So when the concentration increases, you see that the pore size is much smaller, right? So it has bigger pores and it has smaller pores, right? Of course, all these pores will not be uniform, and this is what I'm trying to depict here as well, because it's a polymer settling in to make a semi-solid gel-like structure, but it is enough. We can use this to do a process called electrophoresis. Now, what is electrophoresis? It is using electricity to separate DNA strands of different length. Okay. So together, agarose gel electrophoresis, I hope is clear to you. So what we do in this technique is we make a gel. So let's say this is a gel as I'm seeing it from the top. So it will be thick, slightly. Okay. And on this gel, there will be some indentations known as well. So these are like wells. Wells means groove. In this, you can put your sample, the DNA sample, you can load it here. This whole apparatus is in a liquid, buffer liquid, which can conduct electricity, of course. And what we do at the end is we connect it to a battery, and we connect that battery like this. So there'll be negative end here and a positive end here. Now what will happen is, because this whole setup is in a liquid which is conducting electricity, what will be the flow of where will the electrons move from to? From the negative terminal to positive terminal, right?
Yes.
Now DNA as a molecule, what is the property of DNA? DNA is negatively charged, right? Due to protein.
Yes,
you know that. And if I am establishing an electric field which is negative towards the wells and positive towards the other side, then there will be electromotive force which will take DNA from the negative side to the positive side, right? Because DNA will be attracted towards the positive side. Is that clear?
Yes, sir.
Yes, sir.
So this first lane is always, we call it the reference well or the ladder. We load the ladder here. Ladder is already prepared premix of different sizes of double-stranded DNA, which comes, or you can just buy it. So as this machine will run, your DNA, so let's say this is a 3x or 2x concentration gel. So there will be pore inside the gel, right? So which length of DNA fragments will move the faster and go towards the end faster? Smaller fragments or bigger fragments?
Smaller fragments.
Smaller fragments will move faster. So you will see that smaller the fragment, farther it will move. Because larger fragments will find it difficult to move very fast within that mesh-like network. So this ladder will have different lengths of DNA, and after a fixed amount of time, when you run it, it will spread on the gel like that. And you will get to know that the highest, let's say 10 kb is here, then the next is 8, 6, 4, 5, 3, 2, 1 kb. So the smallest will be the farthest. Now you have loaded 5 samples, let's say. These are all your samples. And you already know that your DNA fragment of interest, let's say it is 3 kb. So this is your DNA fragment of interest. You know it's 3 kb fragment. So after cutting it with restriction enzyme, after treating it with restriction enzyme, if you run the sample, it will have all different fragments, right? So you will see a band here. Let's say in sample 1, this is sample 1, 2, 3, 4, and 5. There are 5 samples. and this is how you find a band. Okay, now can you tell me that in which of the lane you are sure that you have a sample of yours? Which sample contains your DNA of interest?
1 and 5.
Sorry, 1 and 5, right? So you have to look
Yes,
at
sir.
this size. How do you know about this size? Because you have a reference already run. So you can say that, oh, we get a band here, which is for 3 kb. We already know this reference. So this and this might be the DNA of our interest. Okay. You understand, everyone, how gel electrophoresis separates DNA?
Yes.
Yeah.
Yes, sir.
Now what we will do is, after we have separated, we can just isolate, and that isolation can be done by, you can cut this gel piece and elute. That process is called elution. You can elute the DNA which is present. So this DNA, this is DNA for this DNA. You can just take a blade, cut only this piece where you're sure that other sizes of DNA are not available there, and then you elute it out. So this process, write down. Just draw this and write down. Have you drawn this, everyone? You need time. Take a minute and take a minute and let me know when you are done. Yes, Fatima. Can you hear me, Fatima? Is it still lagging? Am I also lagging for others? Donna, Atif?
No, sir.
No?
No,
Okay,
sir.
great. So Fatima, okay, you can rejoin, please.
Done, sir.
Done. Okay. So now write down the separated band of DNA. So this is known as a band of DNA. Okay. The separated band of DNA on the agarose gel. The separated band of DNA on the agarose gel can be cut out and extracted from the gel. Can be cut out and extracted from the gel. Okay. Can be cut out and extracted from the gel piece, from the gel piece. So you just have to take care that you cut as minimum as you want. minimum enough to not let any other contamination come in, and maximum enough to take all the DNA of your interest. Okay. So it can be extracted from the gel piece. This process is known as elution of DNA. Is known as elution of DNA. Now this is the DNA of our interest, which we have now in purified form. And we can now make a cloning vector, a plasmid vector. So is this part clear? Can I go to the next part?
Yes.
Yes. So as we all know that to make a recombinant DNA, we need two things, the DNA of interest and a vector in which we will fit this DNA. So we have our DNA, okay, by this step. Now what we need to do is to design or get a what do you say, cloning vector. Or we can call it plasmid vector, because we do cloning in that, so we call it cloning vector. Because it's a plasmid, it's a circular DNA from which we make the vector for our use, it's called plasmid vector as well. Now write down this question. Many a times this question comes, which is, what are the features that are essential? What are the features that are essential or required to make a cloning vector, or to make a good cloning vector? So what should be the feature present in the vector? What should be the characteristics of the vector? In short, they are asking. This question can come in many forms. How will you design an ideal vector? What should be the properties of a good vector, etc., etc. So basically what they're asking is, what things should be present in your vector which are important? Yes. What? So the following features, you can start writing, the following features should be there. Now what should be the first feature you are interested in based on whatever we have studied till now about DNA and its replication and how we will make cloning? What all properties are required in a vector? Can't think of any? Your vector is going to be a vehicle on which you will load your gene of interest, and you will make a recombinant DNA and send it inside another host to get the gene product. Okay. So what property should be there? For example, if I ask you a question from daily life, if you want to go from your city to another city, that's the closest city to your city, what mode of transport, what should be your vehicle like? What should be an ideal vehicle on which you should do that journey? A bicycle, a car, an aeroplane, or a ship? From your city to another city by land, by road if you are going, what should be an ideal vehicle?
A car?
A car. Why not an aeroplane?
Because it's very short distance and you can just
So
travel by
you
car.
have certain properties in your mind on the basis of which you will select the vehicle, right?
Yes.
So if I say from your country to another country, then what will you choose?
An aeroplane.
And if I say from your block to your friend's house in another block?
A bicycle.
A bicycle, right? So we know what vectors we need to transfer things and what are we transferring. Similarly, you need to transfer your DNA into a host. So what should be the properties that should be there in your vector?
There should be an origin of replication.
Perfect. Great. Let's start with the origin. So there should be a origin of replication. It's also known as ORI. What is an origin of replication, Donna?
The site where the replication of DNA starts.
Starts, right? So this is the sequence where DNA, this is the sequence in that whole DNA from where the replication will begin. And would you like to have just one origin of replication in your plasmid, or you would like to have more?
I'm not
Because
sure.
you would like to have more, because from multiple sites, then simultaneously replication can begin, right? It will take less time and you will make more and more copies. So origin of replication also controls, decides the copy number, how many copies in a given time you can make of your gene. That is also dependent on the origin of replication. So write down. And without origin of replication, no DNA can replicate within a cell. So it is the sequence from where replication begins, and it is also responsible for controlling the copy number of the linked DNA. So whatever DNA is linked to it, which is your DNA of interest, which you want to make clones of, it will control its copy number as well. Copy number is simply number of copies of the DNA. So if you really want to make a lot of copies of a DNA, you would like to have a plasmid vector in which there is a high copy number, origin of replication which supports a high copy number, right? So write down for cloning, origin of replication that supports a high copy number is preferred. Origin of replication that supports a high copy number is preferred. Okay. Good. And what else? So it should have an origin of replication, and that origin of replication should support a high copy number. Great. What else? What else do you need in your plasmid vector? Like seat should be comfortable, engine should support high speed, fuel tank should be bigger if you want to travel for long. All these things you see in a car. So in a vector, what else apart from origin of replication? There should be a selectable marker, right, in your plasmid vector. Because how are you going to know which cells have your plasmid if there is no marker gene? So apart from origin of replication, you need a marker gene that can help you distinguish those cells which have been successfully transformed and has taken up your plasmid from those cells which have not been transformed. So you would only like to continue with the cells which are transformed. You would like to collect them separately and then make them culture and grow. You would not like to waste your time on non-transformants. Okay. So write down. Selectable marker helps in identifying and eliminating the non-transformants. Okay. Non-transformants, those who have not been able to take your DNA. Now, commonly, generally, genes that provide antibiotic resistance, genes for antibiotic resistance are used. So this is an example. The genes that provide antibiotic resistance are used as selectable markers. Can anyone tell me why? How can a gene that provides antibiotic resistance help us select our transformants and separate them from the non-transformants? Donna, any idea?
So could you repeat the question?
The question is, how can a gene for antibiotic resistance, let's say I figured out a gene and that gene provides antibiotic resistance against the antibiotic penicillin. So I want to use that gene as a selectable marker gene when I'm designing my plasmid. So my question is, how does a gene for antibiotic resistance help us like us as a selectable marker? Help us to eliminate the non-transformants?
I'm not sure.
Give it a try. What does antibiotic resistant genes do? I just told you this. What do genes for antibiotic resistance do? They provide resistance against antibiotics. So even in the presence of an antibiotic, the bacteria will not die. Make sense?
Yeah.
So if I'm using this gene as a selectable marker in my plasmid vector, the bacteria that will successfully take up my vector will also take up this gene.
Yes.
They will transcribe and translate this gene, get the protein product, and they will get resistance against penicillin.
Yes.
The other ones which have not taken up my plasmid will have no resistance against penicillin. So after the whole process, if I take total bacteria, I'm not sure how many of them are transformants, how many of them are not transformants. So if I take all the bacteria, put it on a plate where penicillin is present. So all the bacteria who have not taken up the plasmid will die, right?
Yes.
But only my design bacteria who have my plasmid will survive. So don't you think it's a selectable marker which helps me to select the transformants from non-transformants?
Yes.
Everyone understands the logic? Rifat, Atif, Heba, Fatima. Can you hear me, Fatima, now clearly?
Yes, sir.
Is this clear to you?
Yes, sir.
Okay. Good. So that's why genes for antibiotic resistance are used as selectable markers, because they provide us, they allow us to select. Okay. Now we can also use other. I'm giving you one homework here that you have to do. You have to go back and figure out what is insertional inactivation. Insertional inactivation. And how does this is related to a selectable marker? What is insertional inactivation? It's also there in your NCERT. And how is this process related to a selectable marker? Let me also give you a cue that you will read a word called beta galactosidase. And because there is a A's in the name, can you tell me what is this? It's an A's, beta galactosidase, what it can be? It's an enzyme. A's are enzymes, right? Okay, so this is your homework. Everyone has noted down the homework?
Yes, sir.
Yep. Now the third thing, anything else that needs to be in your plasmid vector? Origin of replication to make multiple copies. Selectable marker to identify your transformants from non-transformants. There should be cloning site, a place in the plasmid where you can use the restriction enzyme, where you can insert your gene of interest, right? So cloning site, write down. Cloning site is a sequence where restriction endonuclease can recognize and cleave or cut, because once they will cut, this circular thing will open, and now in between we can fit our gene of interest, right? Now, how many cloning sites do you want ideally in your, how many cuts do you want to make in your circular DNA? So that you can easily insert something inside and close it. If I make one cut, the circular DNA will open, right? If I make two cut, there will be two halves of that circular DNA, correct?
Yes, sir.
Now, these two are useless because I cannot put anything anywhere, right? And close it. So we just need to make one cut so that our DNA of interest can fit in between, and then it will be closed, correct?
Yes.
So ideally, yes, ideally we want that. So that's cloning site, and it also should be there in, okay. Anything else that you can think should be there? I think these three are the most important, okay? Now, when you will be finding insertional inactivation, you will read for beta galactosidase. Also, write one more thing. Look for agrobacterium, again from NCERT. Tumefaciens. What is this? And how is it related to the process? Okay. So this is your cloning vector. It's now ready. So our cloning vector is ready. Plasmid is ready. Now, what is the next step? We're going step by step. So we separated, isolated on gel agarose, eluted, made a cloning vector or plasmid vector.
Could you try?
Sorry for that. Siri gets activated all of a sudden. So then we created the cloning vector or the plasmid vector. Now, what we need to do next? The most important process for which we are doing this whole thing. What do we want to do? We want to get our cloning vector. We have to transfer it inside the cell, right? For which we are doing everything inside a host cell, right?
Yes, sir.
Yes. So now we need to do transformation. Transformation means our gene of interest will go inside a host and will transform the host. and also give the host new phenotypes, or new proteins will be formed inside which we have sent. So transformation of host. And why will any bacterial cell take the plasmid that we want it to take? Right? Why should it take it? Because bacteria have this tendency, but very few naturally occurring bacteria take plasmids or foreign DNA fragments from outside their environment directly into the cell. But we can make even those bacteria which naturally do not take plasmid, we can make them, uptake the plasmid that we have created. Now, how can we do that? For that, we have to make the host competent. So if someone does not want to follow your orders, you are interrogating someone, a criminal, let's say, and is not very willing to be compliant with you, to just support you, what are you going to do? What do we do to get the truth out? Not we, but people who have to deal with them. What do? Simple. It's day-to-day life. You must have seen series, movies. How do you get truth out of someone? That's not a very difficult question, I think. Never seen or heard of interrogations? People. Oh, that's some deliberate silence. Okay, I have no problem. We can just continue that. How to get truth out of someone? Simple, forcefully, right?
Yes.
Yeah, whenever I say something, you say yes, but was it very difficult? If someone is not uttering the truth, you force them to utter the truth, right? And we can do that. We can do the same because these bacteria are not willing to take the plasmid that we have created, but we can't just let them ruin our whole plan and experiment. So we'll force them. We kind of torture them. So what we'll do is, this is biological torture, where actually you subject the cells, the bacterial cells, to a heat shock. So there are many ways to introduce alien DNA into the host cells. So one of the method is microinjections. Okay, you can directly inject your DNA into the nucleus of a cell. Okay, if you can do that, that's called microinjection. Have you heard of a very similar technique I told you about in reproduction chapter, known as intracytoplasmic gamete injection, right? ICGI. Okay, forgot everything. Cool. So microinjection is one. The other, so to write down, to transfer the foreign DNA, which is our gene of interest or our DNA of interest, into a host cell. The following methods can be used. First is microinjection, which is directly injecting the foreign DNA into the host cell, into the nucleus of the host cell. Or what you can do is, this is a very sophisticated method, or there is something called gene gun. You are very right if you understand it. A gene gun is just like a gun where you have very high velocity bullets. So from a gun comes out the bullet, and here what comes out is microparticles of gold or tungsten. Now, why gold and tungsten? Can anyone tell me from your chemistry classes? What is so special about gold and tungsten? No one. Okay. So gold and tungsten both are non-reactive. They are very, very non-reactive, or what do you call non-reactive elements? So they do not react. Sorry?
Inert elements.
Inert, yes. So they're kind of inert. They don't react normally. They're not completely inert actually for chemical reactions, but inside a cell, gold and silver nanoparticles or tungsten nanoparticles or microparticles, they are very inert. They don't do anything. So you take gold and tungsten nanoparticles or microparticles and coat it with your DNA of interest. Okay. Now your DNA is going to be very, very small, smaller than these particles. So many DNAs can be coated, and then you just in a high velocity using a ballistic gun or a gene gun, we call it, you just bombard the bacteria with this the microparticles, and some will enter inside the cell and will take your gene. Now in this method, don't think that the bacteria will be damaged, because these are very, very small particles. They will just, the bacteria will heal up quickly. So these are two methods to transfer forcefully, directly. Another is where we can make the host competent. Now this can be done by heat shock. Okay. So by heat shock method, okay, you heat, so basically you incubate the bacteria on ice, you incubate the cells on ice, then from the ice quickly put it on 42 degrees Celsius. Now 42 degrees Celsius is not very hot to kill the bacteria, but it is from 0 to 42, it's a sudden change in temperature, right? So you keep on 42 for a very brief, just 5 to 10 minutes. So it's on ice, keep it on 42. Then after 5 to 10 minutes, again keep it back on ice. Okay. Now in between, when it was on 42, in the solution, you add your plasmid vector. So what happens? This heat shock. So when things are cold, they are quite compressed. It happens with all our cells as well. The moment there is sudden heat shock, the pores in the cell kind of like open and then again close. So this is how cells behave to heat and cold, right? In general, we also behave to heat and cold like that. So what happens in that meanwhile, the small pores that open are enough to get your plasmid DNA inside. So that's called heat shock method, and it will enable the bacteria to take up the recombinant DNA. So heat shock is ice to 42 degrees and back to ice. Okay, and this is brief, 5 to 10 minutes. That's enough to not kill the bacteria and to also get our plasmid inside successfully. So is it clear, everyone?
Yes, sir.
Yes, sir.
Yeah, so that is how we transform the bacteria and make them or force them to take our plasmid. So that's all the pieces of the puzzle that you needed to know. Starting from the beginning, let's go to the beginning of the chapter. So we started with principles. Okay, we started with bacterial cells. It has a plasmid, so we can use that plasmid. Then we talked about restriction enzymes. We can use the restriction enzyme to cut both the DNA of our interest and the plasmid. And then we can we can separate the cut part through gel electrophoresis. This is another tool. We can make the plasmid vector, and to make the plasmid vector, what we do is we do this here. When we add our gene along with the vector after it is cut, we also introduce an enzyme. Ligase. What do ligase do? You have studied about ligase in it helps to
Join DNA fragments.
yes, ligate or join DNA fragments. Okay. Great. So that's there. That's done. Now, please go through all the techniques once. And now, towards the end, so the last part that we have to do in the chapter is to put whatever we have studied till now, all the techniques together, all the pieces of the puzzle together, and make a flow. Right from the beginning till the end, we have to make a whole process. So the name of the chapter is Principles and Processes. So principles are all, I have explained all the principles, except for two, which is Agrobacterium tumefaciens, and the other one was insertional inactivation. These two I'm giving you as a homework for today's class, and you can come up in the next class with some answers. So I give you 2 to 3 minutes to go through everything in this chapter and ask me if you have any doubt, because next what I'm going to do is we are going to start from the beginning of the process and complete till the gene product is received. So we'll make a full process of recombinant DNA technology. So for that, I would need your participation. So please go through it. Take 2 to 3 minutes. I'll be back in a couple of minutes, okay? Okay, so done, everyone? A quick revision. Am I audible, people?
Yes, sir.
Yeah. Any doubts? Anything which you're finding difficult? If no doubts, then I'll move to the processes part. Now, the process of recombinant DNA technology. Okay, so let's start from you know all the things now, you know all the tools and techniques. So let's start from the scratch. You want to do this on your own. You want to clone a gene of your interest. So where will you begin? You have nothing at hand, you are starting. So where will you begin? Where will Atis begin? See, if you understand this right here in the class, then you won't have to mug it up. So that's why I do this exercise. I can just plainly, define, explain, and go home. Like we are all at home only, but finish the class. But it takes some extra efforts to make it discussive with you people. So take the advantage of this. Where will you begin, Donna? You want to become a biotechnologist? You want to clone a gene, get its protein product. So where will you begin? It's your day one, you entered a lab. What would you like to do?
I didn't understand the question.
The question is, you have a favorite gene in any organism. You choose an organism. Which is your favorite gene in which organism?
Any gene?
Any gene is your favorite gene. In which organism?
Any organism.
So any organism in any gene in any organism you want to clone. So where will you begin is the question.
I'd try and extract the gene.
Exactly. Perfect. So first thing you would like to do, like Donna would like to do, is isolate the gene, right?
Yes.
But can you isolate the gene directly?
No.
What can you isolate? Where is gene present? What is gene?
There's this experiment that I did for gene extraction. Can I just say that?
Yeah, you did experiment for a gene extraction. I'm sure
No,
it
DNA
was not
extraction.
Exactly. So gene is just a small part, a small segment on the whole DNA, right?
Yes.
What you can extract is DNA. So yes, please tell us about your experiment. What did you do? Did you do this in school?
Yes.
Yes. So what did you do, Donna?
We extracted DNA from plant material, banana.
Banana. What was the material? Was it the banana root, shoot, fruit, leaves?
The actual banana, the fruit itself.
The fruit itself, from the pulp.
Yes.
So what did you do? You first took banana, then?
First you peel the banana and then you like make it into a paste.
You mashed the banana.
Yes.
Yes.
And you have to like make it into like really thick paste, like it should not have any
Contamination and
seeds or
water.
anything in it.
Yeah.
And then we put that into a beaker and then we added 20 ml of water, distilled water. And we also added a dropper full of liquid detergent.
Why distilled water? Why not any water?
I'm not sure.
Because any water will have contaminations, bacteria or some other organism, and their DNA will also be extracted, right?
Yes.
You just want to extract banana's DNA, remember?
Yes.
Yeah. So then you added water
And liquid detergent, a dropper full of
liquid detergent. Why? Why liquid detergent? Are you washing the banana before taking the DNA out?
No.
Then what is the first hurdle to reach to DNA? Where is DNA in the banana? Where is DNA in general?
It would
Mm-hmm.
it would be
It would be. Where is DNA in your body? Do you have DNA, Donna?
yes, I do. It's inside the body.
Where?
In the gene.
No, gene is a part of DNA. Gene is just a segment of DNA. So if this is a DNA, from here to here is one gene. Okay. But where is this thing? Where is this long thread in your body? In your nose, eyes, stomach.
In the nucleus, cell nucleus.
In the cell nucleus, right? So in
Yes.
every cell of your body inside the nucleus, there is a gene, there is DNA, right?
Yes.
Perfect. So similarly, in a banana fruit, every cell of the banana fruit will have a DNA inside its nucleus.
Yes.
Now to reach that DNA, you need to first get through the cell wall, then the cell membrane, and then the nuclear membrane. And then you will reach the gene, right?
Yes.
So you have to break this open, and then break this open. So these membranes are broken by detergent. Detergent degrades the membrane. Okay?
Yes.
Cool. So that's why you are adding detergent, because it breaks open the membrane and you can access everything which is present inside the cell. Then what happens?
Then I think you add a certain salt, but I'm not sure what salt it is.
Okay. Why do you add salt?
Again, I'm not sure.
Okay, so you just added a salt because you had some salt, so you thought, okay, let's add salt also.
Yes.
So you added some salt. You also added some other spices, biryani masala, chicken masala.
No.
No, I thought you were making a curry then.
Yeah.
Okay, then
Then
what happened?
you have to mix it, but then make sure that it doesn't foam.
It doesn't foam. Okay.
Yes.
Yeah.
And then you let it sit for a while.
Okay.
And then you add in chilled ethanol. Oh,
You add
yeah,
in
you
chilled ethanol. Okay, that's one interesting thing. Why are you using chilled ethanol?
again, I'm not sure.
Why are you mechanically doing these things if you're not sure? You never asked the teacher, why are you making us add chilled ethanol, first the salt? It's like you're just labors, right? Teacher is asking you to do something, you're just doing it. And that's how students find biology boring, because too many things to do, too many things to remember, but they don't actually ask questions. First thing, you need the organism of your interest, from where you need the gene of your interest. But since you cannot directly take out the gene, you need to take out the whole genome, which is the DNA, the total DNA from the cells. To take the total DNA, you need to burst open the cell. So first you have to make a paste where cells are loose. Then if you want to add some water, you add some water, but make sure that water is not at all contaminated, it's distilled. Then in that water, you add detergent. In labs, detergent is not used because detergent is a very fundamental cheap alternative of enzymes that break the cell open. For example, if you are breaking a plant cell open, you will need a cellulase enzyme because the cell wall of the plant is made up of cellulase. If you want to break open a bacteria, you will need lysozyme. If you want to break open a fungus cell, yeast, suppose your favorite gene is in yeast, So yeast is a fungi, and fungal cell wall is made up of chitin. So you will use chitinase. Because all these enzymes are very, very expensive, so schools don't use it. So they use one alternative, which is a harsher thing that can cleave any membrane. That's detergent. So use detergent to break open cell. Now everything which was present inside the cell comes out open into that solution. Now you want to stabilize certain things. So you don't want protein. Okay. If you don't want protein, because protein is a major component in the cell along with your nucleic acid. There are 4 major biomolecules: carbohydrates, lipids, proteins, and nucleic acids, right? Inside the cell.
Yes, sir.
Proteins are major. You do not want protein contamination when you are taking out DNA. So you add salts because salt makes the protein precipitate. Okay, so protein settles. What remains in the solution is mostly your DNA, some sugars and lipids, and you can also get rid of it by using lipases and amylases and proteases and RNases. So basically all these again are expensive reagents, so you don't use it in school. They just want to show you how a DNA is extracted. But salt is something very cheap and available, so you can actually get rid of proteins. So are you understanding, Donna, why are we doing all these things?
Yes.
Then you don't want it to foam, because if it foams, then a lot of molecules will be in the foam and you will not be able to take it out. Okay. Frothing and foaming brings a lot of air, oxygen inside, and oxygen can also lead to oxidation of many molecules. So mix it, but don't let it foam. It becomes difficult to extract from foams. Then you add chilled ethanol very slowly. And if I'm not wrong, you must have done it by tilting your beaker and then just pouring from the side, right?
Yes, sir.
So this was your already made soup, and ethanol goes from the side. Yes.
I forgot to mention that after you wait for a while, you're supposed to strain it, and then you're supposed to add in the chilled ethanol.
Yes, that straining is to get rid of the pulp simply.
Yes.
We don't need the pulp. We just need the solution, because all the DNA is now in the solution. In fact, everything the cell has is now in the solution. But in that clear solution, when you add ethanol, ethanol makes a layer like this, right? If you agitate the tube, everything is gone. So make sure you don't agitate the tube. Just slowly add ethanol, and it should be chilled, because chilled ethanol, DNA molecules, nucleic acids, precipitate in chilled ethanol. So they come into the chilled ethanol portion. So from this part, your nucleic acid will enter this part and will stay here suspended. Now with the help of a pipette, you will just take this much amount of liquid, which is your ethanol containing your nucleic acids, or you will see, if you leave it there for some time, I think you might not have used a pipette, but what you would have seen, because there is lots and lots of DNA, you would have seen at the interface of it white thread-like depositions, right?
Yes, sir.
That you can put a spool or anything inside, a toothpick, and you can just spool it out, that white thread, and that contains your DNA, they say. So that is millions and billions of DNA threads intertwined together. Of course, it must be having many contamination of lipid and sugar and protein, because you are not using these very expensive reagents to get rid of them. But that's basically fundamentally how you are extracting the DNA. Is that clear?
Yes, sir.
Yeah.
Yes, sir.
Okay, cool. So DNA, when I taught you about DNA packaging, you know that DNA is packed with proteins called histones.
Yes, sir.
Yes. So you can remove that by using proteases, but not necessarily by just adding salt. So when you do a crude extraction like we do in school, which is not specialized extraction, it is not laboratory grade extraction, it contains a lot of impurities, but it's enough to tell you how DNA can be extracted. Okay, in chilled ethanol. So that's called ethanol extraction of DNA. Okay, cool. Then after
Yes.
that, you know other things. You have taken out the DNA. Now you use restriction enzymes to cut it. Then you can separate it using gel electrophoresis. One thing that it is done in industries and labs is amplification of the DNA fragment using PCR. So PCR I'll teach you in the next class. Polymerase chain reaction. And once you have enough number of your DNA fragments, make a recombinant DNA, use a gene gun, ballistic gun, a microinjection, or a heat shock method to put that inside the host bacteria. Then using the selectable marker, clear out your transformants from non-transformants. And finally, when your transformants are there, culture them, grow them in as much of volume as you want. And then your DNA will produce RNA and will produce protein. Once you get lots of protein, you purify the protein and use it for whatever purpose you wanted to use it for. So that's the whole overall process. Is that clear to everyone? So this whole process is there in your NCERT, the last part. Please go today and read about the whole process, and we'll finish it up in the next class. So this chapter will finish in the next class. Okay.
Yes, sir.
Okay, so I'll see you all in the next class. This is time. So I declare the class here. And okay, bye-bye. See you.